So first thing first, guys, for this chapter. So the big portion of this chapter, guys, is macromolecules. Okay. So that's the carbohydrates and lipids and proteins and nucleic acids. Okay. So before that, since you guys get Biochem, this will be a lot easier for me to explain. First, they will ask. They're talking about water. So you guys know about water, H2O. So I don't have to explain that to you guys too much. You know, shape of the water molecules. And also bench shape and hydrogen bonding and all. So I'm not gonna go too crazy there. So they will have a lot of questions, guys, or a lot of like just sidebar stuff about hydrogen bonding. And which you guys should definitely know what it is at this point, right? So hydrogen bond is an intermolecular force, relatively strong. Right, relatively strong. Okay, but the strength of, but these are something to consider as intermolecular forces. Okay, this is something you should know. So in bio, they have some of the IMF. Okay, that they really don't like to use LDF, London dispersion force. They really don't use this guy. Okay, they don't use it. I have not seen LDF in Olympiad bio. Okay. They might say dispersion force sometimes, but again, they only say like LDF. Okay. Sometimes they give you as a van der Waals. Okay. So van der Waals force of attraction. So a lot of I personally think van der Waals force of attraction is being misused in a lot of bio classes. Okay. So van der Waals force of attraction is hydrogen bonds, okay, and like dipole, okay, and also LDF, all combined. Okay, that's what van der Waals force is. But a lot of times, okay, they use van der Waals as LDF. Okay, so a lot of bio classes, guys, just so you know, they use van der Waals force of attraction as LDF. Okay, so go figures, guys. Okay. So there's an ionic interaction. So ionic interaction is not really ionic bonds. Is not really like intermolecular forces, by the way. But kind of funny thing, guys. Okay, in bio, they consider ionic bond relatively weak interaction. Okay, so in chemistry, oh yeah, like Coulombic attraction between plus charge and minus charge, permanent plus minus charge, very strong. Not so much, guys, in bio. Okay, so they think between the charge between plus and minus charges, it's relatively weak. Okay, and they think covalent bond is really strong. Why? I have no idea. Okay, so they say covalent bond, okay, is considered strong. Okay, keep in mind, guys, don't think hydrogen bonding is really strong, guys. Hydrogen bond strength is around 1/20th of covalent bonds. Okay, so if this strength is like 100, like this is what, guys? 5. Okay, hydrogen bond is this weak. Okay. So you'll probably hear these a lot, guys, the terminologies per se, okay, today. Okay. And water can make hydrogen bonding. Okay, so I'm not gonna talk about, guys, like the qualification of hydrogen bonding. That's where you can. Okay. So they also talk about pH for water. So I'm really fast forwarding right now, guys. pH. Okay, you guys know how to get the pH, guys. Negative log of base 10, of the concentration of H plus equals to the pH level, right? You guys also have to know, okay? You guys also have to know, guys, Henderson-Hasselbeck equation. You guys remember that, guys? Henderson-Hasselbeck, guys? You guys, do you remember Henderson-Hasselbeck, guys? So what's Henderson-Hasselbeck, guys? pH is what, guys? pKa plus log of base 10 or concentration of A minus OHA. Conjugate base, concentration over what? The acid, for weak acid. This question came out. Okay, so it's good that you guys have a background in Biochem, okay? And do not forget that for this exam. You guys have to know it. Absolutely no question, okay? They also have an equations about Nernst equation, okay? For Biochem, electrochemistry, Nernst equation. They do not give you the equation. Okay? They do not give you the equation. You gotta figure it out on your own, okay? So do you guys understand why this is a garbage exam? Because okay, so let's say for example, guys, like you never had a chemistry background, okay? They ask these questions. Can you solve it? Absolutely no way. Okay, so there are some questions where you guys need physics, okay? So, I don't know. If you guys want to, I understand, guys, you guys probably the AP Bio next year, but I know some folks from Jericho who's doubled, like AP Physics as well, okay? So, I mean, or whatever, man. Whatever physics you guys have, okay? So, physics questions are there on this exam also as well, okay? So, it's best to have a great exposure to a lot of things for this exam. But by the time you take it, then, I mean, all the electricity stuff in the second semester, so it's not going to really help. But I mean, if you guys want to learn, guys, okay? So that's one thing. You guys know what this question is, right? The buffer zone thing, right? So if you guys have a weak acid and conjugate base at a buffer zone, you can always get what? The pH of solution, right? So definitely not. Okay, I'm not going to write the Learns equation, guys. I mean, the E equation, right? So I don't know when I'm going to teach this, by the way. I mean, so I don't think there's a RT over NF times natural log of Q. So I don't know, guys. I don't know where this will fit in. So in the chapters I'm going to teach, guys, but there will be a question of these, guys. I forgot what's here. Yeah. So yeah, I'm not going to go too crazy with water stuff because like for AP Bio class, AP Bio class, I kind of went over, guys, why like some other concept of water and like solubility, and like the density and all, guys. But I'm not going to do that with you guys, okay? I tell you guys, like the homework, we'll have this. Okay, if you get it wrong, it'll be embarrassing after the AP Chem for the whole year. Okay. All right, so let's just jump right in, guys. Okay. So, and one more thing, guys, with you guys here. Hope I know it's you guys here. So except for this guy. So one thing that I have to make sure with you guys here. Okay, this happens every year. Okay, every year. So what I mean by that is, guys, the questions I'll be giving you guys, okay, homework questions or practice exams and all those guys, it's available online. If you want to find, you can find it. Okay, you know what I mean, guys? So if I give you, oh yeah, today the homework is, okay, 2005 semi-final exam, okay? Like, it doesn't, so the students, okay, they look it up and they just look up all the answers, okay? And you see where I'm going, guys, here? So every student that I taught, okay, I suspect, okay, I don't directly ask them, did you get the answer from somewhere, okay? But I suspect, like, there are some students, like, okay, so they're getting, like, they got 3 wrong. Like, I mean, dude, I get more than 3 wrong, okay? Like, dude, like, if I solve it, I get more than 3 wrong, but they do better than I do, okay? So, and then I think, okay, so something's up here, okay? Either they solved it before, or they have the answer, okay? So, let me tell you one thing. Students who did that, right, never made it. Never made it, okay? So, absolutely never made it. So it doesn't matter if it's Chem Olympiad or Biolympiad. Those who did that never made it, okay? But even for this Chemistry Olympiad past year, there's one student, like he was like the worst student, but he put the work in. Like it was like the last kid. Like he qualified, like because he really put the effort and time in there, okay? I knew some of the kid was looking at the answer key, they don't make it. Absolutely not, okay? So it's hard for me to, like, as an instructor, right? It's hard for me to come out and say, like, oh, you're looking at the answer key? It's very hard for me to say that because I don't want to accuse you guys if you're not doing it. But don't even think about it, okay? Like, don't even try to look for it. Like, dude, we'll go over anyways, okay? So it's a very bad habit, I tell you. Kunde, like, what's really funny, guys, like, the students who, like, even did that and I suspected and then we talked and all, yeah, they still went to Harvard. Like they didn't, like, it doesn't make so that's what I'm saying, guys, it doesn't make a difference. The result of these things, guys, make such a minute difference, okay? So just let you know. That's what I'm saying, like, yeah, it's good for your experience and everything, guys, but, like, I just want to say, guys, okay, so, alright, so make sure you guys understand what I meant by that, okay? Do not, like, cheat even with my work, okay? Give, like, honest work and, with integrity, please. Alright, let's jump right in, guys, macromolecules. So macromolecules, guys. I mean, so there are four things we can talk about. Okay, today: nucleic acids, okay, DNA stuff, okay, proteins, carbohydrates, and lipids. Okay, four cases. Okay, so I'm sure you heard that, guys, like in your like Regents Bio or stuff you just did. Okay, back in time. So let's talk about, guys, DNA. That's the easy one first. Okay, DNA and RNAs, nucleic acids. So keep in mind, guys, in genetics chapter, okay, genetics and you call it evolution. That's a 20% of the test. There's a chapter, molecular genetics. That's when we'll discuss a lot more detail about these guys. Okay, we just have to talk about, structure of DNAs and RNAs and some of the stuff that we need to talk about as part of biochemistry for today. Okay, so let's start with DNAs and RNAs, deoxyribonucleic acid and what? Ribonucleic acids, right? So, and these are something we consider as polymers. Okay, so I'm gonna draw the structure. Let me tell you guys like what you guys have to know. So first thing first, guys, I'm gonna draw the RNA. Okay, so there's carbon here, nitrogen. This is there's a nitrogen here, nitrogen there. Okay, so you'll probably see your textbook, guys. It doesn't matter like where the nitrogen's at, it's fine. Carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, okay? So, and here, this carbon, guys, will have a phosphate group with the oxygen on them, okay? I'm not drawing hydrogen here, okay? So, this is what we call, guys, a nucleotide, okay? It's called nucleotide. Nucleotide is when you have one phosphate, okay, this phosphate group, right? One 5-carbon sugar and one nitrogenous base, okay? That's what we call a nucleotide, right? So now, what's the first thing first, guys? What is the difference between the DNA and the RNA? Okay, DNA is what? Deoxyribonucleic acids, and this is what, ribonucleic acid, right? What's the difference here? It comes down to this guy, the second carbon. Okay, this is carbon number 1, carbon number 2, carbon number 3, 4, and 5. Okay, that's the number of the carbons. Okay, so the second carbon, that's the guy that you have to watch out for. Okay, for an RNA, you see a hydroxyl group. Okay, so yeah. Functional groups. Okay, functional group is what, guys? Like alcohol group, like thiol, like group, SH group, like carboxylic acid, carbonyl group and all. That is something you guys should already know. Okay, after me again. So this RNA, they have on the second carbon OH group. Okay, DNA is missing the oxygen here on the second carbon. That's why it's called deoxy. So on this carbon, this is sugar group, okay? This is a 5-carbon, okay? Sugar group here, okay? It's called ribose. Okay, ribose. So on the 5-carbon ribose, on the second carbon, DNA is missing the oxygen. Okay? So that is one of the easiest ways to determine which one is what. Okay? And also, what's the other difference in DNA and RNA, guys? Based on nucleotide perspective, is what? DNA has what? Thymine. And is a nitrogenous base. Okay, that's DNA. But now RNA has what? Uracil. Yeah. So, okay, we'll talk about it, okay? So that's the very obvious difference, okay, with the DNAs and RNAs, okay? What they're made up of, okay? So now, what is the purpose of a DNA, guys? Yeah, what's the purpose of DNA, guys? DNAs and RNAs, guys, what's their main purpose? Is what? Yeah, like genetic makeup, right? Like, like if you guys have kids, they look like you. Why? Because you guys passed your DNA, okay? So, but just to let you know, right? The primitive, the first, like very early stage of living things, right? Their hereditary material is what? RNA. they had RNAs. Okay, they didn't have a DNA. They had RNAs. Okay, like what do you call that, guys? COVID virus, COVID-19 virus. Okay, what is their genetic makeup? Single-stranded RNA. Okay, do you understand, guys? They got RNAs. Okay, well we have DNA. They got RNA. Okay, so more primitive they are, more likely what their genetic makeup might be RNA. Okay, but why did so again, just point out, I'm not very religious. Okay, so you know. Okay, it's like it's based on a theory. Okay, it takes a very long time, but people believe that what there's evolution. Okay, so we'll talk about, there's a whole chapter on that, guys. So now, if evolution is true, okay, why are we using DNA and not on RNA? That's a question. Okay, that's a question that we'll ask you on the Olympiads. Okay, AP absolutely never they will ask that question anywhere. Okay, because it's too easy, too hard for them. So, two things you have to know. Okay, why we start to use DNA. Okay, because of what? DNA, they are missing one oxygen. Okay, so you gotta understand. So why missing a single oxygen second carbon of the ribose? How does that help? Okay, I mean, why? Like, dude, why does that help us to what? Use DNA and RNA. So you have to understand, guys. You want to, like, I mean, dude, like, think about it, guys. I mean, we're all like Asians, right? You guys are all smart, right? So you know, like, okay, so you guys have a kid. You know, do you guys like want your kid to be like? I mean, you guys want your smartness like go down to your kids, right? Like whatever DNA that you got, you want that to pass down to your kids, right? So DNA, the hereditary material should be what? Change the least. Okay, you do not want it to change. Okay, you do not want your like DNA wherever that's passed down you don't want it to change. Okay, but now for the RNA base, okay. A nucleotide with the oxygen second carbon, what happens? That oxygen. If you have an oxygen, typically what do they do? Yeah, oxygens, right? Like Andy, what do they do? Oxygens. Like in a reaction perspective, like what do they do? Yeah, what do you think, Derek? Oxygens, right?
Electrons.
Huh?
I'm not really sure.
Yeah, the Darcini. It became, you guys, okay? Oxygens, what do they like to do? Yeah, what'd you say?
Something with electrons.
Something with what?
Something with electrons.
What about electrons?
Accept electrons.
Accept electrons.
Oh.
Accept electrons? They're loose as acid or something? Oxygens, what do they have? Yeah, so what do they have, guys? They have a high what? Electronegativity. Right? You can, right? And what? They tend to what? Gain electron. Gain, like take away the electron, right? That's what electronegativity. I mean, we're not doing it chemical, okay? So, oxygens, okay? What do they do? If there are a lot of oxygens, guys, what happens, guys? They're more prone to react, okay? And what? Try to gain electrons. And by having one more oxygen, it makes what? The ribose, what? Stronger oxidizing agent. You understand, guys? So what I'm saying is, compared to, okay, a nucleotide with the oxygen, okay, if you don't have this guy, what happens then? It becomes a weaker oxidizing agent, which prone to what? Less likely to react, because you do not want your hereditary material to react on a daily basis, okay? That's one reason, okay? This is all speculation, by the way, okay? No one knows what happened, like, in 1 billion years. That's why they think, that's one of the reasons why they think we use what? We evolved to use DNA, okay? Less reactivity. So, dude, like, you guys hear this, right? The COVID-19 single-stranded RNA, okay? What do you hear a lot? What do you guys hear? Oh, there's a new variant. You guys hear that? Okay, why? They're RNA, single-stranded RNA, okay? Because why? They're more likely to go under reaction and what? Mutate. Okay, like, oh, we got delta, you know, smaller than like, I don't know, gamma or something, I don't know. That's one reason, okay? And now, listen carefully, okay? This gets a little bit heavy. I don't know about heavy, but let's take a look at this, guys. DNA's have what, guys? DNA's got A and G, and also what? Cytosine and what? Thymine, right? And RNA's have what, guys? A and G, and cytosine and uracil. Okay, so this is a difference also as well. So A and G are what they call purines or purines, whatever how you say it. Okay, and now the other guys, C, T, and U. Okay, so these guys, right? So C, T, and U, like C, T, and U. These guys are what we consider as pyrimidines. Okay. So the difference is, okay, the rings. If you guys like have like, so yeah, how about listen carefully, okay? This is what they call nitrogenous base, okay? You go with this side, right? Nitrogenous base, okay? This is a 5-carbon sugar, the phosphate group, right? So now, if you look at this, okay, if you see like 2 rings like this, those are what we call purines, okay? Which is A and G, okay? Adenine and guanine, okay? So yeah, what do we call this, guys? Adenine. Okay, like guanine. Okay, these are the name of what? Nitrogenous base. Yeah, so you guys have to ask yourself, guys, why do you think they call this a nitrogenous base? Because what? This is basic. They got nitrogen on it, okay? Yeah, well, what do we call this, guys? Yeah, what do we call nitrogen, guys? Nitrogen connected as base, what do we call those guys? Bronsted, the base, okay? They're base, nitrogenous base, okay? So now, why DNA uses thymine, not uracil? That's a question you have to ask yourself. Yeah, why? And how does it help to be more stable, right? That's the question you have to ask yourself, right? So, something to understand, guys, okay? Cytosine, okay, it looks like this, okay? So there's yeah. You guys have to know. I would not push it as far as know how to draw, okay, this. Like draw like cytosine now, okay? Like, but when you see it, you have to know what it is, okay? Oh, this, yeah. AP Bio, your teacher probably doesn't even know how to draw this, okay? So yeah, because it's not needed, okay? You guys have to know what the shape is. Do you understand, guys? So, something like this, okay? So there's a nitrogens like here and like here, something like that. So this is basically, guys, what cytosine looks like cytosine. Okay? I mean, I might not 100% correct, but the important part is where you see this side. Okay? There's some double bonds like here or whatever. Okay, I'm not too whatever. So it's got one ring. So again, if you got one ring like this, okay, these are what we call pyrimidines. Okay, so the important thing is the cytosine got this guy. Okay, this is an amine. So yeah, listen. When they're looking for which of the following is cytosine, okay, you look for that guy. Okay, carbon that has an amine group. Okay, this is called the amine group, right? That is what we call cytosine. Okay. But now, why did we stop using RNA? Okay, or evolve to use DNA, right? Cytosine, they like to go under something we call deamination. Okay, it's called deamination. Yeah, what do you think this is gonna do? Yeah, what do you think this will do? Deamination, what do you think? It removes the amine group. Do you understand, guys? Come on, think about that. What happens, guys? If the cytosine goes into deamination, what happens, guys? It turns into uracil. You know what I'm saying, guys? Yeah, 요게 cytosine spontaneously turns into uracil. Think about it, guys. In your DNA, the cytosine can turn to uracil. You know what I'm saying, guys? So, yeah, because when it does this, our what? Like our cells will know this doesn't belong in our DNA. They fix it. You know what I'm saying, guys? They won't fix this. But if we use RNA where uracil is a what? A base. What happens, guys? How do we know it was a cytosine or uracil? They can't fix it. You know what I'm saying, guys? What I'm saying? So that's the reason why, okay? Speculation, okay? No one knows like exactly why. No, dude, how do we know something that has occurred 500 million years ago? No one knows. 어떻게 알아보지? So, which means what? It's speculating, okay? So what happens? So this is a cytosine, right? So once they remove, what happens, guys? This turns into deaminate. That's what it turns into. Okay? Carbonyl group. Carbon here, and that's what you get. Carbon, oxygen, double oxygen. Okay? That's what you get. That's uracil. But now, what about this guy? What about thymine, guys? What about thymine? What about thymine, guys? Thymine is a little bit different compared to uracil, the shape, okay? I mean, they're very similar, very, very similar, okay? The difference is what? They got one more methyl group on them, right there. Okay, this is called a methyl group, right? So if you look at it, guys, so that's a methyl group right there. So it's easier. So thymine is less reactive, okay? Because of having what? One more hydrophobic, okay, non-polar portion of the base. So you should know. I listen, okay? Like you guys have to understand. You get why this exam is hard. They will ask you a question about these bases. Which of the following is a freaking uracil? If you don't know the shape, guys, it's game over. Okay? That's not something you figure out on the test. Like, I think yours. How do I remember? There's nowhere to go. There's no reference table. Okay? There's nothing. If you don't know it, you don't know it. So that's what's hard. That's what's hard about this exam. That's why you have to spend so much time memorizing these stuff. Okay? 문제가 뭐냐? What's the problem, guys? You memorize today. You turn and you come back in a week, you forget. So that's the problem. Like you gotta maintain, like the knowledge and like memorize every single week. Like it's hard. I get it. Okay, I understand. But that's what I keep on saying. Like know what you're stepping into. So that's probably the second reason why. Okay, uracil is not being used because why? Yeah, so when cytosine does this deamination, okay, turns into uracil, because in your DNA, uracil doesn't belong there, okay, that's why they can go and fix it. But now for them, if we use RNAs, our genome, what happens? We can't fix it because we don't know if it's cytosine or uracil, whatever it was before. That's a speculation that you're using what? DNA. And I trust me, guys, your teacher will not talk about this, okay? Not up to this level. They'll talk about Biochem, but not like stuff I'm doing. Not the shape of like uracil, thymine, absolutely not gonna happen. Because why? If they have a question on this on the AP Bio, guys, they have a structure. They actually give you the amino acid. They give you these bases. Okay. Okay. So now, a couple things more, guys. You gotta read your thing, guys. Okay, please. They're asking that stuff I explained just now. It's not in textbook. The uracil and thymine and all that stuff, it's not in the textbook that I gave you. This is just, you gotta pay attention to it, okay? And if you guys in the recording, guys, like, make sure you guys, like, go back and listen to it. Okay, one more thing, guys, here. So, so that's, this from here to here, guys, okay, is what we call nucleotide, right? From you're here to here, so without the phosphate. What do we call those guys? Nucleoside. Okay. So, have you ever heard something like this? AMP? Have you heard this, guys? Like cyclic AMP, have you heard this, guys? 들어봤어? Have you heard it, guys? Cyclic AMP. This was on, I think, all the AP Chem tests, by the way. Cyclic AMP. Okay? Cyclic what? Adenosine. Okay? Adenosine. It's not adenine. Adenosine monophosphate. Okay? I'm sure you heard of, have you heard of ATP? What's ATP, guys? Adenosine triphosphate. Okay? So what the hell? Yeah. You know what adenine is. Adenine is what, guys? Adenine is the nitrogenous base here. Okay? This is the nitrogenous base. You'll get this is adenine. Okay? But now, when they say adenosine, like these, right? Like A. Adenosine. You know what? They're referring to what? Nucleoside up to here. You understand, guys? Okay? Up to from here to here is what we call, guys, what? Adenosine. The carbon, sugar, and what? Adenine. That's called adenosine. You understand, guys? So if you look at it, guys, okay? Like AMP. Okay? Adenosine monophosphate. Okay? Mono, like M means mono. Okay? Adenosine monophosphate. This is what, guys? Yeah, dude, like, okay, so if you guys have one of this guy, okay, and this is what we call what? So what? AMP, adenosine monophosphate. So now, yeah, the term nucleotide, right? Nucleotide is what? Nucleotide is what? Very generic term. 그지? So it could be what? It could be adenosine monophosphate, which is what? A nucleotide. 그지? And also what? It could be what? Guanosine monophosphate, which is what? This is a guanine. Okay? And like it could be what? Like the other ones, like the pyrimidines have a little weird name, okay? Like uridine, like monophosphate. Okay? So those are the names, okay, for the adenosine. So nucleosides, okay, so these are more generic terms, okay? And like now, like adenosine monophosphate is what? It's a nucleotide. You see that, guys? Yeah, so now, yeah, what about this then? If there's an oxygen there, okay, what do we call this? This is what? Adenosine monophosphate. Yes, right? If you do not have the oxygen, what do you call this, guys? They call it what? You put T there. Deoxy adenosine monophosphate. Okay? They put D there. That's to what? To signal, okay, so there's what? Is it a base for a DNA or the RNA? Okay? So if the D's not there, means what? Yeah, it's what? It's got the oxygen in the carbon. Yeah, you see them what? What about C? Cyclic AMP? What? Yeah, cyclic AMP. What do they do? Cyclic AMP. Yeah, what do they do? Cyclic AMP.
Respiration.
I will just rest. Calm down. Respiration. Cyclic AMP known as what we call a lot of times cyclic AMP known as secondary messengers. Okay. So there's a whole chapter. Yeah, I tell you guys have to listen into that AP Chem class. Okay. So what was the difference, guys? So the AMP, right? This is adenosine monophosphate. What's the difference, guys? This right here will bond to the third carbon right there. Covalent bond. So this is like a little cycle. Okay. That's why it's called cyclic AMP. If you break that, it becomes AMP. And I'm sure you heard, like, dude, like, okay, like ATP, right? Adenosine triphosphate. What do you think of? You got what? You got 3 of these what? Phosphate groups. Okay. That's ATP, right? ATP adenosine triphosphates. And again, these guys have what, guys? Negative charge here. So now, if you look at it, guys, DNA and RNA, they're what? Made from these guys, right? And DNA stands for what, guys? Deoxy. Okay, like ribonucleic acid. Got deoxyribonucleic acid. So this is deoxyribonucleic acid. DNA, right? Yeah, but now you ask yourself, guys, okay? Yeah, where is the acid? Acid all these where's the acid guy? Yeah, okay, where is the acid? So DNA, so this is there's only one, right? Yeah, where is an acid? Yeah, acid what do you mean? So like this is a base, right? This is a base, right? As we discussed, nitrogenous base. Okay, this side is basic. Yeah, where's the acidic? Where's the acidic side? Yeah, well, this side. This is what? Phosphoric acid. They lost H plus, which means that this is minus charge. Okay, that's why it's called an acid. So DNA is what, guys? Relatively negatively charged. Okay. So this side is acid, that side is basic. What do we call in bio, even in chem, a molecule that has both acidic and basic? What do we call that, guys, in chemistry or bio? Yeah.
Amphotericity.
Oh, because you're amphoteric. Yeah, exactly. It's called amphoterism. But now in bio, they don't like to use the term. This term, guys, in bio, a lot of terminology, they like bio and chem, they don't really like to use the same term. They call it, guys, all the amphiprotic. Okay, amphiprotic. That's a term they use also. Okay, amphoterism. Okay, also sometimes called amphiprotic. Okay, that's what you can be both acid and base as a single molecule. Okay. Keep in mind, guys, okay, there's a much heavier section about the structure and like whatever, about the DNAs and RNAs, guys, in the future chapter. Okay, I'm going to the next stuff. Like protein synthesis and all that stuff, guys, by the way. But what about, guys? So now let's talk about, guys, a polymerization of these things. Okay. So just to like make sure you guys understand, guys, here. So very simple. I'm going to draw this very simple. Okay. Nitrogen. Okay. And then there's carbon here, oxygen, phosphate, oxygen, oxygen. Okay, that's it's one base. Okay. And now there's a second base I'm going to draw here. And then there's carbon here, oxygen, phosphate, oxygen. It's a second base. Okay. So now, if you look at it, guys, here, so you can kind of see where this is going, right? So if you look at it, so this is if you look at this is a fifth carbon, okay, and this is a carbon number 1, carbon number 2, carbon number 3, carbon number 4, okay. So if they're chaining up, okay, turns into what happens? Turns into a polymer. What happens, guys? The third carbon is going to bump to the oxygen on the phosphate, okay? And this is a fifth carbon, this third carbon. They're gonna, they're gonna have a long chain of this. So that's why they say, guys, okay, it goes from the 5 end here, okay, 5, 3, 5, 3. So this is what we call, guys, what? They say, okay, going from the 5 end of a 3 end. This is a 5 to a 3 end strand of DNA, okay? That's where these terms like 5 to 3. I'm sure, like, a couple of years ago, I'm sure you probably heard double-stranded DNA. There's a 5 side, there's 3 side. And this is a 5 and 3 side. I'm sure you heard this, double-stranded, right? So what does this 5 and 3, the numbers represent? It's the numbering of the carbon. Okay? 5 first, 3, 5, 3, 5, 3. It goes down like that, okay, on this strand, okay? And now, just so you know, this bond here, okay, double bond here, whatever, okay? this bond between two of these bases, guys, and this is what we call phospho diester bond. So if they ever ask you guys, what's the bond between the two bases of the nucleotide bases, guys? What's the answer again? Phosphodiester bond, okay? That's the functional group that you have to know. So I tell you guys, like, Olympiad Bio, they do not go absolutely crazy, okay, with chemistry questions, okay? This is not Olympiad Chem, okay? Olympiad Chem, like, you guys have to know mechanism behind how to create, like, the bond. Like, they don't go that far, okay? Like, organic chem perspective. And I'm sure you guys all know this, right? The base pairing, like A pairs with what, guys? T, right? And G pairs with C. You guys know this, right? Do you guys know this, guys? And one thing that you have to know, and you have to know even for the homework, is this. A and T, they create only 2 hydrogen bonds, okay? While C and G can what? Make 3 hydrogen bonds. Okay? So now, if there's a greater C and G combination, then what happens, guys? It has what? It's harder to separate the strands of the DNA, okay? Like this, they call it like dissolving, or even what? Like melting, like, you know, like separation. So think about it, guys. If there's more C and G, which means they have a triple hydrogen bonding, okay, that's holding them together. It's harder to they take more energy to separate the 2 strands of DNA, okay. Vice versa, if there's more A and T, it takes less effort to separate them. Yeah. Any questions, guys? Let me know. So again, like there's a lot more to this, okay? But as far as this chapter goes, I think, and the questions that's on the homework, I think this is all you have to really know, okay? I mean, of course, like you might say, oh yeah, this is, I don't get it, but we'll discuss that, okay? Any questions, guys? All right, let's go to guys. Let's go to carbohydrates, guys, which is the sugar. So I really want to finish this, guys, because the whole thing, because this is it's not that hard, I think, because you're coming from chem background a little bit. So carbohydrates. So whenever you talk about carbohydrate, what do you guys know? It has a carbon and what? Carbon and hydrogen and oxygen, right? Like, you know. So what's the typical ratio, guys, here? Typical ratio is what, guys? Like carbon and oxygen, I mean oxygen and hydrogen is a ratio 2 to 1, okay? So this is typically like, this will be X here, that's Y there, okay? So in X and Y, like the numbers are relatively similar, okay? So, you know, so the typical formula is what, guys? Like C, like 10, like H, like what, like 22 or something, whatever. Like O, 11, something like that, okay? So it's like a little bit like what? Like 1 to 1 ratio, okay? And now, they don't have, they can, but a lot of times they do not have a nitrogen and phosphorus, okay? They got only these 3. And carbohydrate, right? What do we typically use them for? Is what? Usually what these are for? A short-term energy source, right? So, you know, like obvious one is what, guys? The one thing that they'll ask a lot, if they do ask, is glucose. Okay? Of course, right? Glucose. You heard that here and there, right? Which is C6H12O6. 근데 I tell you guys, okay? Even for glucose, same glucose, but there are many different forms and different name for this as well. Okay, so it'd be nice and simple in the AP Bio. Oh yeah, glucose. Next chapter. No, like dude, it's there's a lot more to it. Okay, There's an alpha glucose, beta glucose, L glucose, D glucose, aldose, dextrose, they got like different name for the same crap, okay? So you gotta understand what they mean by this, okay? And the structure of the glucose as well, guys, okay? Is it a linear structure, is it a ring structure? Like what the hell's the difference here, okay? Like what's the alpha and beta difference in the structure perspective? So there's a lot of questions on glucose, first of all, okay? So glucose, guys, first thing first, guys, okay? If you draw glucose, this is what it looks like, linearly. So it's 1, 2, 3, 4, 5, 6, right? And now there's a OH group here, OH group here, OH, OH, OH. Okay. Hydrogen, hydrogen, hydrogen. So I'm not gonna okay. This is carbon number 1, carbon number 2, carbon number 3, 4, 5, and 6. Okay, that's glucose. And now you guys took chem. Okay, AP Chem, you guys are smart kids from Jericho, or so I heard that you guys are like top kids in Jericho, right? Dude, I don't know about that, but dude, like, okay, yeah. So, organic chem, okay? 요게, if they give you this, okay, they call this a D glucose. Okay, in case you're like, you probably will see guys on these questions, they'll say, oh, D glucose, okay? What D stands for is a dex, okay, which means like, it means the right side, okay, the right side. So, okay, in terms of, like, I know AP Chem is not too strong on organic chem, I get it, okay? But now, because of the chiral center, so again, like chiral center, I'm not gonna go too crazy right now, guys. For the chiral center stuff, guys, because of the chiral center of this carbon, okay, here, like could be here, it could be a lot of different places here. It's got a mirror image of this molecule. Okay, so if you put a mirror there, there's a mirror image of this guy. Okay, I'm not gonna draw it. That's called what? L, left and right. Okay, leva, something, I don't know what the Latin word, guys. There's an L and D. They look the same. Go do that. What do you think, guys? They're like left and right hand. Yeah, left and right hand, yeah, dude, they look about the same, but they're not exactly the same, right? You cannot overlap them. Okay, exactly. Okay, so that's something we call, guys, something we call enantiomers. Okay, that's what we call L and D. Okay, in case you wonder what they are. Okay, like they say, a lot of times they say, sometimes on the choices on the Olympiad bio exam, they say a D glucose, okay, or L glucose. It means what? There's a different variation. So yeah, the left hand, glucose, right hand glucose, okay, something like that. Those are called enantiomers, okay. So now, and now, glucose, right? This is like, they're not the most stable form as a linear structure. Why? Why? Why is it not so stable as a linear structure? Why? MD, what do you think? Why would this not be so stable as a linear structure? Why?
There's a lot of bonding sites.
What do you think?
Not sure, maybe because it's not actually.
Yeah, if there's a single bond, what do you guys know? Yeah, well, what do you think, guys? Single bond, what happens, guys? They can rotate around the axis. Okay? So they're gonna turn. This thing turns. I mean, it's not like straight like this. I mean, they will turn on every single axis, on this bond, that bond, that bond, that. They're gonna turn around. So yeah, if they start to turn and all that stuff, what do you guys know? It will have what? Higher entropy. Okay? Yeah, if they have a higher entropy, what do you guys know? They have what, guys? They have a what? Higher free energy. Okay? Which means what? Unstable. Do you understand, guys? So what do they do, guys? They rather will like to have this in a ring structure that you've probably seen somewhere. Okay, it's glucose molecule. So they actually turn it into what, guys? Like something like this. I really want you guys to, I don't know, man, like, I've seen questions where you have to know this is a glucose, okay? So, just so you know, this is why this is hard, guys, okay? You gotta know the structures, man, I tell you. Let's see what happens here. So first carbon, second carbon, third, okay, so, yeah, so carbon, 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 CH2. This is glucose, by the way, okay? So this will ring like, so it will turn to a ring. Rings cannot rotate on the axis, okay? Which means what? Or stable. So, but what you have to understand is this. Here, okay? So now, if you look at this, guys, so you have an OH group here, H here, OH here, H there, OH here, H there. Keep in mind, okay? These are not flat shaped, okay? So man, this is not flat. You gotta understand that, okay? They're not like flat shaped, okay? So they're like, you know, like chair shaped, and like, so we don't have to get there, okay? Cyclohexane, you know that, guys? So, but now, yeah, this is a carbon number 1, carbon number 2, carbon number 3, carbon number 4, 5, and 6. That's a number of carbon. Okay, like these fold into this. You know what I'm saying, guys? But now, it's hard, you're going to think about this is important, okay? There is something we call alpha or beta glucose. Okay, alpha or beta. It all depends on the first carbon. The carbon C1, right? That carbon will decide whether it'll be what? Beta or alpha glucose. Okay? And now, if you have stuff like, if you have like this, where the OH is on the bottom like this, okay? This is something we call alpha glucose. So if you look at Papa, OH down here, OH down here, okay? Yeah, well, what do we call those guys? We call this what? Cis. Cis, right? Yeah, do you remember this, guys, from chem? If they're on the same side, what we call this what? The cis. Remember that, guys? If the X is on this side, what are we? We call this what? What do we call this, guys? What do we call ND? If they're on this and that side, what do we call this? Huh? 아유, yeah, what do you think, Darcy? It's called what, guys? Trends. Oh, yes. Yeah, this is AP Chem, dude. You also 나왔는데 there's a question like this in AP Chem. SAT Chem, they had this too. You know. Yeah. Cis and trans. What do we call this guy? What do we call this what, guys? Cis and trans is something we call geometric isomers. Yes. 똑똑하다고요. 야, 똑똑하다. You guys, someone said you guys are smart, guys. Yeah, geometric isomers. I can't teach you like every little stuff like this, okay? So something like cis and trans called geometric isomers, okay? So enantiomer is a mirror image. Geometric isomer is what? Cis and trans. Those are examples of geometric isomers. So these are all part of what, guys? Isomers. Same formula, different structures, okay? So if you look at it right here, okay? The OH group on the C1 and C2, they're what? Facing the same side. Okay? That's why I'm calling cis. Do you know what I'm saying, guys? Okay? And if it's a cis, it's what? Alpha. Glucose. But now, if this is not what it is, if it's flipped, okay? If the OH is on the top, H on the bottom, okay? 이거 뭐야? Trans. This is what? This becomes a beta glucose, okay? Alpha and beta, these are examples of what we call the anomers, okay? Anomers. Just write that. Anomer for alpha and beta glucose. Okay, why is it alpha and beta, guys? Because alpha, like first one, second one. Okay, there's no like deep meaning to this. So so yeah, so you guys understand, guys, what it means by L-glucose, D-glucose, alpha-glucose, beta-glucose, okay? That's based on how they fold, okay? One thing to understand, guys, beta is more stable, by the way, okay? Beta is more stable because why? Why? We don't want to why. Oxygens are very electronegative. They want to be far from each other, okay? So trans is what, guys? They're far away from each other. That's why it's more stable, okay? So beta is more or less, guys, more stable form compared to the alpha. You get why this is so important, guys? Is if, okay, so now, yeah, glucose is, yeah, like there are other, other, carbohydrates we can talk about, but this is actually one of the ones that they have the most, of course, right? Glucose molecules, right? 80% of the entire thing is for glucose. So, why this is like, even like on the Olympiad exams, like easy questions they have these, okay? So this is a glucose, okay, which sometimes they call this monosaccharides, right? Okay, one single molecule like this, and now they can chain up, okay, just like DNA base, right? They can chain up, 1, 2, 3, 4, 5. They can chain up, right? So first thing first, let's kind of understand, guys. Okay, let's look at alpha first. Okay, alpha. There's OH group on the bottom. H up here. So now, if there's a second glucose molecule, so now, yeah, you got to know these guys. Okay, 2 glucose molecules chain up turns into maltose. Okay, this is something in the reading you guys have to know. Okay, 2 glucose turns into maltose. Okay, so maltose disaccharide, right? So basically, what happens is, guys, here. So I'm not gonna do the whole thing here. So this is a hydrogen here, OH there. And this is a hydrogen here, OH there. So now, they will be able to chain up, right? And in bio, they call this what? Dehydration synthesis. Very easy stuff that you probably heard like sometime in your Regents bio stuff, right? So in chemistry, what do they call this, guys? I'm not gonna ask you guys. It's called condensation reaction, okay? So the terminology is, they're the same thing. Different terminology, but the same thing. Condensation reaction or dehydration synthesis is what? They kick out the water molecule and what? Create a covalent bond. So OH group on the C1 goes out, hydrogen will on the C4 goes out. What happens, guys? H2O pops out. What happens, guys? They will create a covalent bond. Okay. This bond that was created between the C1 and C4 of the adjacent carbohydrate. We call this what, guys? Alpha 1,4 glycosidic linkage. Okay. It's called alpha. Yeah, alpha. This is alpha glucose, right? 1,4 glycosidic linkage. This is alpha bond. Okay. So this is disaccharide, which is what we call maltose. Okay. They got a long chain. Like if you turn this into a long chain, one straight long chain of this, what happens, guys? Turns into what? Turns into starch. Starch is a bigger term, guys. Starch can be what, guys? Amylose and amylopectins. Okay. So which means they can turn like a long chain. This is for the plant. Okay, plant. If the plant turn them into a long chain, what happens? Turns into what? Starch. Like starch is a very generic term, which can be what specifically segmented into what amylose or amylopectins. Okay. And we do have the enzyme to break this down. Okay, we have the enzyme to break these guys down. Okay, so we have the enzyme called amylase, and what? Break it down. Okay, and in liver cell, what happens, guys? We can do the same thing, which the alpha 1,4 glycoside linkage. Okay, what happens? We can turn into what? Glycogens. Okay. So glycogen, amylose, and amylopectin, they're about the same thing. Okay, except like, it depends on the branching. Okay, which I'm not going to talk about today. So now, why we talk about this right now, guys? You guys understand a beta glucose. Okay. So the beta glucose molecule, they can't really do this as how the alpha is doing. So if you look at it, guys, as we know, beta is this. Okay, the OH group and H group is what? They're like sort of what? Now the other way here. Do you see that, guys? So the one was down here, the alpha was down here, right? So they can easily just chain up and that was easy, right? But they can't do that. So meaning guys, if there is another beta glucose, like this, right? So the OH group is down here. So they can't really make a bond because they're not like directly lined up. What happens, guys? Today, this guy needs to be flipped. You know what I'm saying, guys? Okay, it's got to be flipped. Okay, so glucose, alpha glucose as well, guys, nicely chaining up like this, right? But this guy's what? You gotta flip this guy like this. Okay, so every other one is what being flipped. Okay, so because of what, guys? That what? Being like a anomer of each other. Okay. So now, if you look at this guy, so it's flipped version, right? So I gotta flip. Okay, flip. If you flip it, carbon here, oxygen here. Okay. So now there's so this right here, same thing. I mean, so but now if you look at it here, what happens? This is what we call a beta 1,4 glycosidic linkage. So if and then you can make what a long chain of this guy, okay? What does this turn into, guys? Yeah, what? If you create a long chain of this guy, what happens, guys? This turns into cellulose. Cellulose. So as I said, yeah, what did I say, guys? Beta is more stable form, right? There's more of beta, okay? Which means what? There's more of what? Easier to create cellulose compared to what? Starch for the plants. And we do not have an enzyme to break this one. We can break down alpha, but we cannot break down beta, okay? So these are what, like, yeah, so cellulose is what, guys? A source of a cell wall, like a cell wall for the plant, right? And like, you know, like the heavy bulk of the trees, right? Like, you know, if you look at trees, man, like, they're tough, right? Like, they're what? Because what? They got what? A lot of cellulose, okay, build up. Off topic, right? Off topic, right? Where do you think? What do you think is a source of fossil fuel? What do you think, guys? Fossil fuel. Source of fossil fuel, guys? What do you think?
Carbon.
Huh?
Carbon.
Carbon.
Yeah,
What do you think? Yeah, what do you think, Darcy? Fossil fuel source. What do you think? So, you get like, let me tell you guys, okay? So, like, what happens like today, right? If tree falls down, right? What happens? What happens? What do you think? Yeah, what happens? If tree falls down, what happens? It decomposes. If it gets rotten and they go away, they go bad. Why? Yeah, what makes them go bad? What do you think? So decomposers, okay, such as bacteria and fungi. How do they eat that like tree? Because why? They have what? Enzyme to break this down. Okay? We don't, they do. Okay, so they decompose what? Yeah, they can turn this into what? A glucose. You know? So, well, good, great, right? But now, for when the plant was first able to produce cell wall, be able to do this, okay? For millions of years, okay, there is nothing that will be able to break down cellulose because they don't have the enzyme to break down the beta glucose linkage, okay? So if trees fell, it will not decompose for millions of years because there's nothing you can eat that guy, okay? So now think about, imagine guys, right? Trees fall for millions of years and nothing can touch it. What happens, guys? Yeah, just get piled up, right? And then what happens, guys? It's deep in the ground, and they get heat up, and what? Turns into fuel. So cellulose is what? Is a source of. So undecomposed plant cellulose is a source of fossil fuel. Okay? Can we make that now? We can't. So just like what, guys? Like the boys, what do you think, guys? Like you guys probably heard, guys, what? Yeah, have you ever heard, guys, open hypothesis, okay, and Miller-Urey experiment? Have you heard that, guys? Yeah, well, in the pyramid atmosphere, okay, They got like ammonia, they got like H2S, like sulfur, they got like methane gas. There's this little spark, and what happens? They will have to create what amino acids. That's a Miller-Urey experiment, right? Like that's open hypothesis. Can they do that today? Okay, but can they do that today? They can't, because why? We have too much what? Too much O2 gas in the atmosphere. Same thing. If the plane falls down, oh yeah, let's try to like create like fuel today. Can they do that? No, because what? They already have so many bacteria and fungi. If the tree falls down, they're gonna eat them all. Okay, so it was a one-time thing, during the evolution perspective, but they can't do it anymore. Yeah, that's basically what it is, guys. Okay, and it's cellulose. So just keep in mind this is on your homework question. Okay, so like glycosylated stuff. Shouldn't say this. And sometimes, guys, if you look at this. They have different names for like the same stuff as I mentioned, guys. Yeah. If you look at this, right? This is carbonyl group. Okay. As organic compounds, what is it? Is this a carboxylic acid? Is it an amine? What the hell is this? Wait, what is it? What is this, guys? Yeah, well, this is an aldehyde. You understand, guys? Aldehyde. Right? So this is what? Sometimes they call this what? Aldose. Okay, glucose is a part of what? As an example of an aldose. It's a sugar, okay, but that's an aldehyde. Some, like, fructose, for example, okay, they're what? They are ketose, okay, because what? They're ketones. So, like, they have a lot of these different names, guys, okay? Like dextrose, okay, because it's what? It's actually what? This is basically what? Like D-glucose, right? Then they call it dextrose. So they got like it's the same thing, a different name, like a little more broader terminology, okay? So we'll see. You'll see some of the terms here, okay? Like I don't know what the homework, but I don't know. If you guys go to college and maybe take biochemistry, guys, then like, you'll probably hear these terms. So keep in mind, guys, like carbohydrate stuff, they can't really ask a lot of stuff on them, but little details, okay? And you have to understand, at least you have to understand, guys, the shape of like glucose molecule in a ring structure. At this one, I want you to know it, but I don't know. It's textbook they have it, okay? So you gotta memorize them, okay? I mean, there's lots to memorize, but this is not in the front line here, actually. You gotta know the nitron space guy. You have to know uracil, cytosine, and like uracil, cytosine, and thymine. You gotta know what it is. So you gotta memorize these things. Okay, but like you memorize today, what's the chance you're gonna memorize? It's gonna be fresh until February. You gotta keep reminding yourselves. That's what it comes down to, guys. Yeah. That's why like there's a review sheet that I have to give it to you guys. Yeah. But like practice exams, you gotta keep on going back. That's where the problem is. Yeah. Any questions there, guys? All right, so let's talk about, guys, the most important thing, guys. Just jump right in, which is a protein. If I don't finish the whole thing by today, guys, you still have to read and do the homework, guys. Okay? So I'll tell you which one to read. So the protein is. I'll do the important one. So there's one more, lipids, and you gotta talk about the enzymatic reactions, guys. But probably lipids is not too much of the questions that you guys have to do. Enzymes, you might have to do the readings. It's like 5 pages. All right, so protein, guys. Protein, as you guys know, it's one of the macromolecules. So this is made up of what we call the amino acids, right? How many amino acids do we have, guys? There are 20 amino acids. Okay. So you have to know. So listen carefully, okay? You have to know and memorize amino acids. No question, guys. 20 of them, you have to memorize them all. Okay. Just to let you know, guys. I don't memorize them. Okay. I don't have to memorize. I'm not a test, guys, by the way. So I don't memorize them, by the way. So I know a few. Okay. But for you guys, at the end of the day, you have to memorize them. Okay, so for last year, the student that I told you guys, who took the test, guys, on 2021, like I asked her, like, because it was online, I kind of felt bad, but like I asked her to make a printout and put it on her desk when she took a test. I mean, so, with 20 amino acids, I don't think you do that this time, guys. By the way, you gotta memorize it. Okay. So amino acid, guys, basically the shape looks like this. Okay. That's the bare bone shape of amino acid, hydrogen here. Okay. Every single one looks like the same. Looks the same. So amino, okay, because why? This is what? Amine group, right? This is amine group. Okay, right? This is what? Carboxylic acid. Carboxyl acid. That's why it's called what? Amino acid. Just like guys, okay? And now if you look at this, how about I go? Look at this, guys, okay? Basic, acidic. What do we call this, guys, again? Amphiprotic or what? Amphipathic again. So amphiprotic or what? Amphiprotic, okay? Amphiprotic or amphiprotic. Basic, acidic. And now R, right? This one. This is sometimes they call this a side chain group. Side chains. Okay? So there are 10 hydrophobic. Hydrophobic, like what it means by hydrophobic, guys? Non-polar. Okay, non-polar groups. There are 5 hydrophilic, like polar. Okay. And I think there are 3 and 2. Okay, I think 3 like basic or something, and 3 is acidic. So acidic is minus charge, and basic is plus charge. So that's how the breakdown is. Okay, 10, 5, 3, 2. Yeah, you gotta memorize it. Like you gotta know like glutamic acid. That's acidic amino acid. Okay, like you gotta memorize that. So the list is like in the textbook. The earlier, the better. Okay, like for example, glycine is what? It only has like hydrogen there. Okay, that's glycine. Okay, so and like what? Phenylalanine, like there's some like crazy ones there. Okay, so maybe not the structure, but glycine is hydrophobic, something like that. You understand, guys? Okay, glutamic acid, acidic. Okay, phenylalanine, maybe basic. I'm not sure which one. Okay, so something like that. Okay, like proline, hydrophobic. Like I know a few. Okay, but not everything. So I guarantee, like 진짜로, I guarantee. Easy question, okay? So 이거 진짜 every year they have a question like this. Every single year, like you gotta memorize the amino acids. Okay, there's every single year one question on this. Okay, based on the memorization of the amino acids. And that's what makes them like. And now, if you look at this, guys, this carbon, okay, again is what? It's a chiral carbon, chiral center. So again, sometimes you see guys like L, like glycine, or L-leucine. I'm sorry, glycine can be, okay, and then D, like you see this again, okay, L and D, okay, right hand, left side, okay, because they have a chiral center carbon, but they don't like to play with this too much, okay. So yeah. So that's a basic, structure of the amino acids. Okay. The AP bio, right? They got a lot of questions on this one. The protein, a lot of them. If there's a question on the biochem, it's on this one. Okay. And now, if they're chaining up, guys, the 2 amino acids are chained up. So there's one more. Okay, here, right? Okay, the 2 of them. So now what happens, guys? So this OH and H, okay, will be going out as dehydration synthesis, right? What happens, guys? They will be turning into a single bond. Okay? Now, there's a name for this bond. So again, the DNA is what? Phosphodiester bond, right? The glucose, like the carbohydrates of what, guys? Glycosidic linkage. 얘는 뭐 같아? What do you think, guys? It's what? Peptide bond. Okay. It's called peptide bond. Okay. So now, if you look at it, guys, right? There's a long chain of the amino acids, right? So if you look at this, guys, let's assume, guys, right? There are this is a long chain of amino acid, okay? 요거, this side is like the end of one amino acid, and that's the other end, right? Long chain. 요거, this side is called the N terminus, N terminal. Okay, because there's an nitrogen there, and this side is what we call the C terminal. Okay, I guess it's C. The last one is there. Okay, so there's always N terminal and C terminal of the protein. Okay, because every time, like one end should be nitrogen, the other should be carbon. Okay. I said it, guys. Earlier, you memorized amino acids, the better it is for you guys. Okay. And again, like I don't memorize it. Okay, like I forget it anyways. Okay, so but you guys, so sorry guys, but you guys have to memorize it, guys. Okay. I tell you guys, your teachers will never ever will ask you to memorize it. Okay, ever, absolutely never. Okay. Okay, so now let's take a look at guys. Okay, so what is really important about this, guys, is the structure of a protein. Okay, so in terms of a structured protein, there's what we call primary structure of the protein, secondary structure of the protein, tertiary and quaternary structure of proteins. Okay, there's a primary, secondary, tertiary, quaternary. Sorry about that, guys. So, I mean, guys, dude, you guys know this, right? Primary structure is what? Sequence of the amino acid. Sequence, like which one comes first? Methionine, glycine, leucine, proline, glutamic acid, glutamine, like the sequence, okay? Which one comes first, second, third, fourth, whatever? That's a primary structure. So primary structure is what? The sequence of the amino acids, okay? There are 20 amino acids and what? Yeah, like which one comes first, second, and third, okay? So the primary structure of the protein is determined by what? Yeah, what decides the primary structure? What, like, is it random? I'm gonna put this guy first, this guy second and third. Here you go. You know, is it like that, guys? When your mom or dad, whoever that cooks, right? Today I'm gonna just mix all these together and just give to him. No, there's a standard protocol to making food, right? Like, you have to boil the water first, you put the whatever, you know, like whatever. So the structure of the primary structure, which is the sequence, comes from what? It comes from DNA. DNA's got them, okay? That's the reason why, yeah, think about it, guys. If your hereditary material is what? RNA and mutated tomorrow, what happens, guys? Your offspring might not able to produce what? A functioning protein. Do you understand what I'm saying? Okay? Like cystic fibrosis, for example, okay? It's all hereditary stuff. So primary structure, DNA, okay? Sequence. What else? What about secondary, guys? 요거부터 이제 약간 좀 secondary. What about secondary, guys? Secondary structure, you gotta know this, okay? It's either alpha helix or a beta. They call it pleated sheets. Okay? Why is it called alpha and beta, guys? Because this was found first, this was found second. And there's no meaning of the alpha and beta, okay? Like there's no like, beta, the structure, no. It's just how it is. They found first, second, okay? So what the hell is that? What's alpha helix, what's beta sheet? So basically what happens is like there's a long chain of amino acids, right? So they turn into like a DNA, like, you know, turns into like a helical shape. Similar thing, right? So it turns into like a helical shape like this, okay? That's called the alpha helix. And now, to do this, okay, the main force behind this is what? Hydrogen bonding. 요거 진짜, they ask this a lot. Okay, oh yeah, hydrogen bonding, where does it exist? Primary, secondary, tertiary, quaternary, okay? Primary structure, they don't have hydrogen bonding, but secondary, they do, okay? So now, how do they create this? Alpha helix, like where, hydrogen bonding where? So, if you look at it, guys, here, there's an amino acid here, right? here. I'm sorry. There's your carbon here. The side chain R. This is a double bond O. You know, like there's a nitrogen there. So it's like repeated of these guys, right? Like, you know, amino acids, like all repeated. So now what happens is, on the other side here, there's a nitrogen there, okay, with hydrogen. There's a carbon here, there's a carbon there, okay, double bond, and there's an R group and stuff like that, okay? So now, I'm sorry, swapped, doesn't matter. But now, hydrogen bond, what's hydrogen bond, guys? So, so this is, I don't wanna, so not that it's important. Hydrogen bond is what? If you have a hydrogen that is bonded to a nitrogen, okay, example, right? This hydrogen qualifies for hydrogen bonding, okay? When what? When there's what? Oxygen or fluorine or another nitrogen present, okay? You look at this right here, they can what? Form hydrogen bonding. Do you understand, guys? So the hydrogen on the amine, okay, is being hydrogen bonded to what? The oxygen of the carbonyl. Do you understand, guys? That's hydrogen bonding. And that is the force that's responsible for what? Alpha helix. Okay? Throughout this entire thing. So this is where, guys, like, you gotta understand, okay? Does the side chain matter for this? The side chain, the side chain is what? The R group, right? Does R group matter for this? Not so much, because why? Every amino acid has what? Carboxyl, carbonyl, and what? The amine. Okay, so it's not so much of what? The amino acid specific. Okay, unless we're talking about what? Unless we're talking about proline. Okay, amino acid proline. I don't know how to say it. Okay, chlorine, proline, whatever. They're a little bit different. Okay, unless you talk about this guy. Okay. Yeah, keep in mind, okay? One thing to mention, guys, okay? There are 20 amino acids, right? 2 of them have sulfur. Okay, so like amino acids, what, guys? They got nitrogen, what? Oxygen, hydrogen, carbon, and some of them have sulfur. Okay. Sorry, guys. This is a friend. Just went to Korea. I just called because I just landed. Sorry, guys. All right, so hello. Yeah, listen carefully. There are 2 amino acid with sulfur. Okay, you gotta memorize. Like, so dude, like every time you gotta memorize, okay? Cysteine, and methionine. Okay, these guys have sulfur. Okay, none of them else will do. And they, as you can see, they don't have phosphorus on them. Okay, cysteine and methionine, they got sulfur in there. Okay. And proline, their structure is a little bit different, okay? So you know, that's why they might have impact on that shape, by the way. Okay, that's alpha helix. What's a beta sheet, guys? Beta sheet is what? So if these strands of the amino acids, they like run parallel to each other like this, parallel, whatever, okay? So the hydrogen bonding can be created, guys, what? Like this, okay? Between the strands of like the parallel lines. That's what we call, so when they create like a sheet of the amino acid, that's what we call the beta pleated sheets. Okay? Same understanding. The oxygen on the carbonyl, hydrogen on the amine group, they create hydrogen bonding. Okay? So secondary structure, it's not so unique, okay, to a particular amino acids, okay? So the amino acid, a lot of the proteins can have what? These 2, okay? Some like very short, like, you know, structures like proteins, they don't even have this. But a lot of the long ones, big ones have these, okay? And now, 3, this is the most important thing, by the way, okay? which is the tertiary structure. So let's talk about guys, right? Like, I guess you guys have like a few minutes left, but tertiary, right? Yeah, like I tell you guys, like you guys have to join the Saturday class, by the way, okay? Because I'm sorry, I talked about this already. Some of the things could have been covered by that class, okay? So I don't have to repeat this. So the time is the essence here, guys. Like, so I don't know. I'll talk to them, guys. I prefer you guys to be in person, okay? But if okay, like I'll have you guys just on the Zoom, okay? So that Saturday, were you guys busy 9 o'clock Saturday? What do you do?
Oh, wait, no, I'm pretty sorry.
Okay, so because I did this already, so I don't want to repeat like again. So because like it's about the same, okay? Like except like when you guys come, then we can talk about other stuff that's not part of them. Okay, so this is like it's sort of double work for me, okay? So so that's why I said like because I thought like I don't know they said you guys took AP Bio, so I'm like okay, okay, so and now they said like you guys are pretty. I mean I don't know about that yet, okay? So but you guys are like top kids in Jericho or something. Yeah, like okay.
All the kids in Jericho are the same.
Yeah.
What?
Everyone's the same.
Everyone says they're the top kid?
Everyone's top. Everyone's the best.
Okay. Okay. So like, I was like, okay, like, oh yeah, like I have some good kids this year, like okay, like. All right, proteins, right? So protein, guys, you can understand, okay? What's the function of this macromolecule? So DNA and RNA, they're for the hereditary materials, right? And carbohydrates for what? Like short-term energy. Protein, what about them, guys? What do you think? Well, a lot of proteins are what? Proteins are, there are two different types. Globular proteins, I'm not going to write it, okay, I don't have time. And what? Structural protein. Structural protein and globular protein, okay? Structural protein can be like collagen, okay? Your like skin and hair, okay? So they're for structural purposes, right? Okay? They don't do anything except for what? Just maintain the structure. Globular protein is more kind of fit into this, okay? Globular protein is what, guys? Something like what, guys? Enzymes, right? Enzymes, okay? Which what? Catalyzes a particular specific reaction, okay? So now, you gotta say, right? Bio, right? Bio is what? Like, so you guys did chem, right? Like catalytic reactions, right? So it's a little bit different when you consider bio. So chemistry is where, oh yeah, if you had a catalyst, the reaction speeds up, right? Oh yeah, it's faster, and then things occur faster, and it's all good, But bio, guys, is more about control. They want you to speed up a particular reaction when you need to, and slow it down whenever it's needed to as well, okay? Control, okay? So a reaction in bio should never occur on its own, but it should be what, guys? More of a control. There must be a trigger that, okay, so you go and react, okay, you gotta stop now. So there's a lot of these inhibitions and activation that happens, okay? So there's an enzyme for every reaction that's out there, which catalyze reactions. So there are thousands and thousands and thousands of these protein, globular protein, which are the enzymes, right? So if you think about it, right? So then, there's a reaction 1 and 2. They should have a different enzymes, okay? But how do they know they're different stuff? Like how do they do that? It's what's important about these proteins. What? Their specificity, okay? Like the tertiary structure gives them what? Specificity. Like special shape, like a very unique shape, okay? That's what tertiary structure gives them, okay? It's got that unique shape or what? It catalyzes only a particular reaction when needed, okay? So it doesn't catalyze the other guy, it only does this guy, okay? That's what tertiary structure is for. 이게 가장 중요한 거. This is the most important thing, okay? So how do they do this? So what gives them this specificity is tertiary structure. So basically what happens is what, guys? So tertiary structure is basically the more or less, guys, it occurs by the specifics given as what we call the protein folate. Yeah, protein folate. Okay, so the long chain, the alpha helix, beta sheet, and long chain is what? They're folding into a certain place. Okay, how do they know though? Okay, how do they know where to like how do they know what to fold? So this is where the primary structure comes into play. So where the sequence comes in. So basically what happens what guys? What will decide the specifics to what guys? The side chains. Okay, the side chains. So the structure, the sequence, right? So the first one is methionine, the second one is glycine. You know, what's the difference between those amino acids? The side chain groups, the R groups, right? And based on the R group interactions, they will what? Yeah, they'll interact with each other and what? Folds into a specific shape. So there's more to come. This is I can't like explain this in 5 minutes here. This is the one portion of it. So this is what I want you to do. Okay. There will be a homework assigned to you guys. Okay, this is an online thing. You guys should kind of know how to go, where to go, and I'll send the email for this, guys. Okay, I don't know, probably tomorrow, if not Thursday. Okay, so yeah, so there's an online site that you have to go and do the test. I think there are like 30 something questions, like 37 questions. I'm not sure. It goes from again 2014 to 2004 to 2014. Every question then of this stuff. Okay, so what we're missing right now is the rest of these guys, okay, which you guys definitely have to know to do the homework. And also lipids, okay? And also the enzymes, that's what's missing for you guys, okay, to do the homework. So regardless, okay, so we gotta go. Like, dude, 27 weeks left, guys. I can't like, oh, let's discuss the rest of it next week, and like, no, I can't do it. So what you guys have to do is, you guys have to somehow listen into the Saturday class, okay? The AP Bio class, okay? And then, okay, you guys have to do the readings, okay? And then you guys have to do the homework, okay? So there's 30, so that we have to follow there's a schedule, okay? We have to finish everything by October. So the reading, I'll assign you guys have the textbook. I'll assign you what to read, okay? And then I'll send you an email with instructions and stuff like that by tomorrow, okay? All right, guys. And I'll try to upload this and share it, guys, okay?